this year's laureates of the Nobel Prize in Physiology or Medicine. I much look forward to learning more about your findings concerning peripheral immune tolerance. I will now give the floor to Professor Gunilla Carlsson-Hedestam, member of the Nobel Assembly at Karolinska Institutet, to introduce this year's laureates. Thank you.
Esteemed Nobel laureates, dear colleagues, friends, and all of you who have joined us here today in Aula Medica. On behalf of the Nobel Assembly at the Karolinska Institute, it's my great pleasure to introduce this year's Nobel laureates, Dr. Shimon Sakaguchi, Dr. Mary Brunkow, and Dr. Fred Ramsdell. The three laureates have been awarded the 2025 Nobel Prize in Physiology or Medicine for their discoveries concerning peripheral immune tolerance. The Nobel lectures are the start and the scientific highlight of the Nobel Week. The findings about which we are about to hear are fundamental to our understanding about how immune defenses are kept in check. The identification of molecularly defined regulatory T cells launched an expansive research field of relevance to many areas of medicine: autoimmunity, organ transplantation, cancer biology, and neuroimmunology. We live in a world full of microorganisms, and we depend on them to maintain a healthy ecosystem on our planet and inside our bodies. To protect us from potentially disease-causing infections, we have evolved an amazing immune system that has equipped us with highly effective layers of control. Immune responses are powerful and they must be regulated. To fine-tune these strong responses, many feedback regulation mechanisms exist. While all living organisms have evolved some sort of defenses, the adaptive immune response is particular to vertebrates. Central to adaptive immune system are T and B cells, each of which express a unique antigen receptor on its surface. These receptors are generated through combinatorial gene rearrangements, providing us with an extremely diverse repertoire of receptors that mediate recognition of all potential intruders that might infect us. But how can the adaptive immune system distinguish between foreign structures and our own tissues? How is immune tolerance achieved? This question has puzzled immunologists for a long time, and one critical answer was provided with the description of central tolerance, a process whereby self-reactive T cells are deleted in the thymus, preventing potentially harmful cells from entering circulations. But this did not explain everything, since some self-reactive cells are known to escape thymic selection. Many groups were interested in understanding the mechanisms behind immune regulation. But the experimental toolbox was quite limited during the 1960s and 70s, and although it gradually improved, it was nothing like today's easy access to monoclonal antibodies and single-cell sequencing methods and other tools that greatly help today's researchers in defining cellular subpopulations. The discoveries for which the 2025 Nobel Prize is awarded is a story of two research fields coming together. The first involved findings in cellular immunology, and the second in molecular genetics. When the pieces were put together, the findings resulted in the definition of regulatory T cells, cells that really stand out among other immune cells, since instead of attacking, they dampen immune reactions. The story starts with Dr. Sakaguchi, who, after completing his medical studies at Kyoto University, was determined to follow up on previous observations that removal of the thymus from 3-day-old mice resulted in severe autoimmunity. By performing a series of cell fractionation and transfer experiments, he could show that CD4 T cells that also express the cell surface marker CD25 display regulatory functions. The results were published in a landmark paper in 1995. Much earlier, in the 1940s, at the Oak Ridge National Laboratory in Tennessee, researchers had noticed a spontaneous mutant mouse strain, which they named scurfy. The male scurfy mice developed severe autoimmunity, while the female mice did not. This suggested that the mutation was located on the X chromosome. This mouse intrigued the personnel to such an extent that the scurfy mice were kept going for half a century. This in itself perhaps deserves a prize. They realize that this may be of great scientific value one day. But it was not until the 1990s that the scurfy mice finally were subject to detailed molecular and genetic characterization by Dr. Brunkow and Dr. Ramsdell, who at the time were active at the company Celltep Science. The severe autoimmune phenotype that the scurfy mice displayed suggested to them that if they could only find the mutation, this might provide really crucial clues to understanding immune regulation. As so often when great discoveries are made, it's about identifying an important question and pursuing it. As we will soon hear from the laureates themselves, this work led to the identification of FOXP3, a gene that encodes the transcription factor that programs T cells into becoming regulatory T cells. Soon after the discovery of FOXP3, Dr. Brunkow and Dr. Ramsdell showed that young boys with the severe autoimmune disorder IPEX also had a mutation in the FOXP3 gene, demonstrating its relevance to human disease. These landmark papers were published in 2001. Following these discoveries, Sakaguchi's team connected the two findings when he showed that FOXP3 was selectively expressed in CD4 and CD25 positive T cells, while Ramsdell team showed that the regulatory T cells were absent in the scurfy mice. With these pivotal results, the floodgates opened, and immunologists all over the world began to study regulatory T cells. The aim was, of course, to understand their biology and to develop clinical applications. On behalf of the Nobel Assembly, it's my great pleasure to introduce Dr. Shimon Sakaguchi, who will be our first speaker this afternoon. Shimon Sakaguchi was born in Nagahama, Japan, and he received his medical degree from Kyoto University's medical school in 1976. He then received his PhD from the same university in 1983. Following some years as a medical resident and about 10 years as a researcher in the US, he developed this field. He was active both at the Johns Hopkins University, also as a visiting scientist at Stanford University, and the last 2 years of his US tenure were he was active at the Scripps Research Institute in La Jolla. After returning to Japan in 1991, he worked as an investigator for the Japan Science and Technology Agency, and he was a professor at the Department of Pathology at the Institute for Frontier Medical Sciences at Kyoto University, where he was also the director between 2007 and 2011. Since 2011, he is a professor at the Department of Experimental Immunology at Osaka University, where he's also the vice director since 2012. Please join me in welcoming Dr. Sakaguchi to the stage to deliver the first Nobel Lecture in Physiology and Medicine 2025.
Thank you, Professor Hidestam, for a kind introduction. So, Excellencies, ladies and gentlemen, it is my great honor and pleasure to receive this year's Nobel Prize in Physiology or Medicine, together with Mary Brunkow and Fred Ramsdell. I'm grateful to the Nobel Committee and Nobel Assembly at the Karolinska Institute for selecting us for the award. So today, I'd like to talk about the role of regulatory T cells in immune tolerance. As shown on this slide, the immune system it protects our body from invading microbes. It has unique properties. T and B cells, they have a reactivity to diverse array of antigens. They have a high antigen specificity and immunological memory, and also a potent mechanism of antigen elimination by the antibodies and the T cells, helper T cells and killer T cells. And also the immune system, normally it doesn't react with self-antigens. At the turn of the century, Paul Ehrlich, he coined the term the horror autotoxics. It means the horror of self-toxicity. He thought that normally the immune system doesn't react with our body. But at the same time, he stated that there must be certain regulatory contrivances which protect ourselves from autoimmunity. Since then, several mechanisms of immune tolerance have been proposed. One is deletion. So immature T cells or B cells, when they recognize self-antigen, they may be deleted, kind of apoptosis, deleted from the immune system. Second idea is inactivation. So usually T cells need not only antigen recognition, but also co-stimulation. Just on recognizing antigen, then these lymphocytes become inactivated. In immunology terms, anergy. And third mechanism is suppression. Even normal individuals, they may possess a potentially hazardous self-reactive lymphocyte, but their activation and expansion is controlled by another T cell population. Now we call them regulatory T cells. So the key question is, which mechanism, if it doesn't work, whether it can cause autoimmune disease or not. So autoimmune diseases, it is estimated that approximately 8% of the population is afflicted with some sort of autoimmune disease. For example, multiple sclerosis in the nervous system, thyroiditis or gastritis, or the adrenal gland, or Langerhans islets in the pancreas. All these, the autoimmune disease in these organs, it's a very popular autoimmune disease in humans. And then sometimes ovaries or testes, they are also affected, causing infertility in female and male. And also, there are systemic autoimmune diseases, such as SLE, systemic lupus erythematosus, and rheumatoid arthritis. And then one feature of these autoimmune diseases, that is overlapping. So multiple organs are frequently affected. For example, type 1 diabetes, thyroiditis, and then gastritis. So from that point of view, there is an interesting experimental system developed by two Japanese pathologists, Yasuaki Nishizuka and Teruo Sakakura. They found that if you remove the thymus around day 3 after birth, this neonatal thymectomy can cause various autoimmune diseases. For example, here, strain A, B, C, strain A, when they are removed of the thymus around day 3 after birth, the 30% developed the autoimmune gastritis, and around 30% autoimmune ovulitis, autoimmune disease in the ovaries. But interestingly, day 0 thymectomy, during the 24 hours after birth, almost nothing happened. The day 7 thymectomy, also nothing happened. If you change the strain to strain B, and now day 3 thymectomy caused the ovulitis, about 90%, with some other autoimmune diseases. In contrast, strain C, if you remove the thymus around day 3, nothing happened. So this means that the day of thymectomy is critical. And also, if you change the genetic background of the mice, you see other autoimmune diseases with different frequencies or resistance to autoimmunity. So how neonatal thymectomy should cause autoimmune disease? So what we found is that if you inoculate normal T cells from adult mice, especially CD4 T cells, after neonatal thymectomy, then you can prevent disease development. So how? Our hypothesis was the thymus is producing T cells from gestational days on. So day 0, mice were born. So day 3 thymectomy, it means perhaps before thymectomy, perhaps the thymus has already produced the autoimmune T cells. And then around day 3, they start to generate the kind of autoimmune suppressive T cells. Now we call them regulatory T cells. So day 0 thymectomy, no T cells, no autoimmunity. Day 7 thymectomy, already enough number of regulatory T cells in the periphery, so no autoimmunity. Now the question is, how we can separate these two populations, regulatory T cells and autoimmune T cells, from normal adult mice? And then whether if we remove regulatory T cells directly from normal adult mice, can we induce autoimmune disease or not? And so here, normal mice in the spleen, lots of T cells. And then we started to dissect the T cell population in splenic T cells by the expression of various cell surface markers. So how to remove this? It's an antibody plus complement, and then the antibody react with this certain molecule, these molecules, and then they kill these cells. And then after that treatment, if you transfer to the T cell deficient mice, and then if you see the autoimmune disease, then this means that removed population may be suppressive, and then inhibiting the autoimmune disease development. So first we were successful with a CD5 molecule. And then 5 years later, Fiona Powley and Dan Mason at Oxford University in the UK, they showed that CD40RB is another marker for that population. And the question was then, by removing the smaller number of T cells, and then if we have the broader or more serious autoimmune diseases, that is a more specific marker for this population. So, and then we end up with the CD25. So here, this is a flow cytometry, and each dot means each lymphocyte. So here, a CD25 positive and a CD4 positive. This population, when it is removed by antibody, anti-CD25 antibody plus complement, and then this population is gone. And then if you transfer to T cell deficient mice. So now you see various autoimmune diseases. So for T cell to transfer to T cell deficient mice, nothing happened. And then if you transfer CD25 minus cells, 100% of mice developed gastritis, autoimmune disease in stomach, ovaries, ovulitis, 100%, thyroiditis, 70%, and then also other autoimmune, in other organs, salivary gland, adrenal gland, islet, and type 1 diabetes, kidney, glomerulonephritis, as seen in SLE, or joint the arthritis, as seen in human rheumatoid arthritis. And importantly, if you add back the removed population, CD25 positive cells, now you can suppress disease development. So in vitro, we can demonstrate their suppressive activity. So in the spleen, CD25 positive cells, around 10% of CD4 positive cells. And also in the thymus, 5 to 10% of CD4 positive cells. And then they are not, if you remove them, CD25 positive cells, now you can enhance the proliferation of responder T cells, CD25 minus cells. And more importantly, if you co-culture with CD25 positive Treg here, and then responder T cells, now Treg suppress the proliferation of this population in a dose-dependent fashion. And so Treg also suppress CD8 positive cells. Similarly, in the thymus, CD25 positive here, regulatory T cells can suppress the proliferation of other lymphocytes. So these findings suggest that the thymus produces Tregs as a functionally distinct and the competent T cell population. So here in the thymus, the Treg differentiate into mature population. So compared with other T cells, it may take longer for them to become mature within the thymus. And that is the reason that their differentiation or generation is a bit delayed. And so neonatal thymectomy around day 3, this means that by day 3 thymectomy, a bit the Treg generation is delayed. So you can selectively or relatively reduce Treg, and you can cause autoimmune disease. And second, thymic Treg population links central and peripheral tolerance. So the T cells react with the ubiquitous or abundant self-antigen, and they'll be deleted by a clonal negative selection or clonal deletion. And some of them with intermediate affinity for self-antigens, somehow they differentiate into Treg. And then low affinity ones, they can escape the negative selection and go to the periphery. And some of them self-reactive T cells, when you remove Treg, they become activated and cause autoimmune disease. So central tolerance and the peripheral tolerance, somehow they can be connected. And then also, whatever it is, genetically or environmentally induced alteration in Treg number or function can be causative of autoimmune disease. So we have shown that the various environmental causes, for example, certain virus or certain chemical or the X irradiation, all can reduce regulatory T cells and then cause autoimmune disease. Like the cancer development, the viral carcinogenesis or chemical or the physical carcinogenesis, just the affecting the oncogene or tumor suppressor genes. So, but in autoimmune disease, not gene, at the cellular level, this population is affected, and you see autoimmune disease. And then various molecules, if you look up the knockout mice, they develop autoimmune disease. Now the question is whether this kind of mechanism is also operating in humans or not. And then the very interesting findings were made by the Mary Brunkow and Fred Ramsdell in 2001. So they positionally cloned the genes responsible for scurfy mouse, very severe systemic autoimmune disease. And then immediately they showed that this gene, they named FOXP3, a transcription factor. It is responsible for human rare autoimmune disease, IPEX syndrome. So IPEX syndrome, the one gene defect can cause the autoimmune disease in various organs, and especially type 1 diabetes and inflammatory bowel disease. And so after when milk feeding start, and then intestinal microbes develop, and the commensal microbe develop, and then you see the start of inflammatory bowel disease. And allergy, when the baby start to take food, immediately they show food allergy. So we got interested in this gene because the disease picture is so similar to what we saw after Treg depletion. So later on, we made an antibody against FOXP3, and then we found that normally 10% of CD4 T cells express FOXP3, but in Scurvy mice, no such cells. And now we can stain regulatory T cells with FOXP3 and then CD25. So what we found is that the majority of FOXP3 positive cells, they are CD25 positive in the spleen, also in the thymus. So mouse study and the human IPEX syndrome, it can be connected. So 3 groups, including Haas, and then the Sasha Rudinsky's group in Seattle, and the Fred Ramsdell group in Seattle, all 3 groups published this finding that FOXP3 is specifically expressed by regulatory T cells. So what does this mean? Here, the potentially hazardous T lymphocytes are present in every healthy individual, but under dominant suppression by Treg. So it's amazing that the FOXP3 mutation, the 80 to 90% of kids, the affected patients, they develop type 1 diabetes. This means that whatever HLA they might have, the Treg deletion or Treg defect can cause the type 1 diabetes and other autoimmune diseases. So, and then second, the border between self and non-self is not fixed, but movable by controlling the number and function of Treg. So self, non-self discrimination, the immunologists that prefer that word, but a border between self and non-self, it is not fixed. You can move this by the increasing Treg or reducing Treg. So this means that autoimmune disease can be treated or tumor immunity evoked and transplantation tolerance established by controlling the number and function of Treg. So this is the tumor immunity. Here, if you reduce the number of Treg, and then this is a T cell deficient mice transferred with CD24 T cells or Treg depleted cell population. And if you put in the inoculate tumor cells, without Treg depletion, a tumor become bigger and bigger, and then all the mice died of tumor progression. But here, without Treg, first tumor become bigger and then rejected. And after rejection, if you immunize with 10 times the number of tumor cells, they are immediately rejected. So this means that, like a measles infection in viral infection, we can make the normal mice specifically immune to the tumor cells. How about transplantation? If you raise this border up, and then you can induce transplantation tolerance. So again, T cell deficient mice. So without T cells, they can accept the skin graft from other strains of mice. And then if you inoculate, so 100% accepted. And if you transfer the normal T cells, strain A, and then you see the rejection happens. So within a month, all the grafts rejected. But if you first transfer the Treg, and 1 week before, then Treg are recruited to the graft site and they expand, and then suppress the rejection. So the rejection become delayed, and 30% become permanently tolerant. And if you increase the number of Treg, now more than 70% become permanently tolerant. So without using immunosuppressive drug, just expanding naturally present Treg, we can induce transplantation tolerance. So with the discovery of CD25 as a marker, cell surface marker for Treg, and FOXP3 as a specific marker in the nucleus, now we can define Treg as distinct cellular entity. And then publication on Treg now rapidly increased, and then especially it allowed or enabled the deciphering the molecular basis of Treg development and function. And also, it has enabled clinical application of Tregs. So, what's the molecular basis of Treg function? So here, the Shohei Hori in my lab at that time, so he expressed the FOXP3 gene in normal T cells by retroviral vector. And then without Treg, and you transfer Treg depleted cells into T cell deficient mice, you'll see the severe autoimmune gastritis and colitis, IBD, inflammatory bowel disease. But if you co-transfer with FOXP3 transduced normal T cells, now they can suppress the disease development. So FOXP3 gene can confer a suppressive activity on normal T cells. So how? FOXP3 controls hundreds of genes, either activate or depress. For example, CD25 become activated, CTLA4 activated, IL-2 interferon gamma depressed. The question is, among the FOXP3 control genes, which one is important for this suppressive activity? So this means that which gene, when it become deficient, whether such mice develop the severe autoimmune disease as seen in FOXP3 mutation as scurvy mice. There are not so many. One group is IL-2 and then IL-2 receptor. So CD25 is the IL-2 receptor alpha chain. And then another one is CTLA4. So one cardinal feature of Treg, that is they are constitutively expressing the high affinity IL-2 receptor composed of alpha, beta, gamma. Already, when they are produced in the thymus, they are already expressing the high affinity IL-2 receptor. And FOXP3 repress IL-2 production. So normal T cells, when they are activated, they express the IL-2 receptor and also produce IL-2. And then IL-2 bind to the receptor, and then T cells become, they expand. But on Treg, they don't produce IL-2. That is the cardinal feature of Treg. So it means that they are highly dependent on exogenous IL-2 for survival. So if you neutralize IL-2, just injecting anti-IL-2 neutralizing antibody, then immediately you see the reduction of the Treg in the spleen, also in the thymus. And then you see the very severe autoimmune diseases. So in this case, it's an NOD mice, the type 1 diabetes prone, genetically prone, and then the diabetes become more severe. And the thyroiditis, the thyroid is almost completely destroyed, and the salivary gland also destroyed. And then how about the CTLA4? So Kajsa Wing, she was a postdoc from Sweden, and she showed that when you make the CTLA4 Treg specific and CTLA4 deficient mice, conditional knockout mice. So here, FOXP3 positive cells, they are expressing CTLA4. And then if you make a conditional knockout mice, FOXP3 positive cells here, but then they are not expressing CTLA4. And these mice spontaneously developed very severe autoimmune diseases, such as autoimmune myocarditis. And they show the infiltration to the cardiac muscle, and then cardiomegaly, and then they die by heart failure. And if you look at the IgE in the circulation, the concentration is several hundred-fold higher, meaning that they are prone to develop the allergy. And then CTLA-4, as you may know, anti-CTLA-4 antibody now under clinical use, ipilimumab. So, so these mice die soon. So we transfer the spleen cells from these mice to normal T-cell deficient mice, and then inoculated tumor cells. And then red lines, conditional knockout mice, they rejected the tumors, but control mice, all of them died of tumor progression. So just the CTLA-4 deficiency in Treg, it is very similar to what we see after removing Treg. So CTLA-4 is the important molecule for Treg function. So this is our current understanding of how the various molecules expressed by Treg, how they are involved in immune suppression. So here, CD80, CTLA-4 interact with CD80-86. So they are called co-stimulatory molecule. For responder T cells, antigen recognition plus co-stimulation, then they become activated and expand. So if Treg reduce the expression of CD80-86 by trogocytosis and then transendocytosis, so just snatch out the CD80-86 from the cell surface of antigen presenting cells. And then also the soluble CTLA-4, they block the residual CD80-86. So very efficiently, the Treg block the co-stimulatory signal. And second, the PD-L1, which is again, it's a good target for cancer immunotherapy. So PD-L1 is normally associated with CD80. So CD80 become reduced. Now free PD-L1 increase, and then interact with PD-1 positive T cells. Namely, Treg can suppress activated PD-1 positive T cells. And if the Treg further differentiate, they also secrete immunosuppressive cytokines, IL-10 or TGF-beta. And one of the key targets is antigen-presenting cells, their expression of co-stimulatory molecules and MSC. Plus, the high-affinity IL-2 receptor, Treg, they absorb IL-2 from the surroundings and then depriving IL-2 from responder T cells. So with these findings that the various molecules, key molecules are now expressed. And then the next question is the autoimmune disease of, the monogenic autoimmune diseases, FOXP3, CD25, CTLA-4, all develop very severe autoimmunity. And now they are called T-regopathy, and then list of the T-regopathy is now expanding. And how about the autoimmune diseases, general, polygenic one, which afflict around 8% of the population. So we know that the single nucleotide, the polymorphism, SNP, it can contribute to the genetic disease susceptibility. So we analyzed the autoimmune SNPs. So they are expressed by many of them, 60% expressed by the immune cells, especially CD4 T cells. And then the question was whether this the variation is the loss of function effect on Treg, or gain of function for helper or other CD4 T cells. And the conclusion was many of them are controlling the Treg development. And so the enhancer region of Treg signature genes, lots of Treg, the autoimmune SNPs, and they locate and then contribute to genetic susceptibility to autoimmune disease. Now, how about treatment? So you can expand Treg because they are highly expressing the high affinity IL-2 receptor constitutively. So just low dose IL-2 can selectively expand Treg. It's not enough for expansion of other T cells. So you can expand Treg and then strengthen immune tolerance. And second approach is the Treg they are present in the circulation. So take them out and then expand them and then transfer back to the patient. The third one is this is what we are working on. How we can convert antigen-specific T cells into Treg in vivo or in vitro. And so the effector cells, they are converted to Treg, FOXP3 expression plus Treg-specific epigenetic changes together. And now we can have a very stable Treg. And if you transfer back to the patient, you can suppress the disease development. So now Tregs are ready to go to the clinic. So as I told you, by expanding Treg or strengthening their function, we can treat autoimmune disease, allergy, or immunopathological diseases such as IBD, and organ transplantation. And then other, for example, heteromaternal tolerance to prevent abortion. Then you can expand Treg. On the other hand, by reducing their function or expanding attenuating their suppressive function, you can enhance the immune responses, such as in tumor immunity. So. This is, so I'd like to acknowledge the contribution by many people. First of all, I'd like to thank my wife, Noriko Sakaguchi. She's a partner in my life and also collaborator in research. And the mentors, as I already told you, Dr. Nishizuka and Sakakura, they introduced to me how to induce autoimmunity by neonatal thymectomy. And Toshitada Takahashi, he taught me how to dissect or separate T cell population by antibody. And I have been collaborating with many people, including my colleagues and students and collaborators. I have mentioned some of them, but others also they contributed the progress of our Treg research. And funding source, the funding source in Japan and also in the United States. I greatly appreciate their support for our research. Thank you very much for your attention.
It is now my great pleasure to introduce Dr. Mary Brunkow, our second laureate for this afternoon. Mary Brunkow was born in Portland, Oregon, where she earned a Bachelor of Science degree in Cell and Molecular Biology from the University of Washington. She then transitioned to Princeton University, where she completed a PhD degree in Molecular Biology in 1991. She then moved to the Samuel Lunenfield Research Institute in Toronto for postdoctoral training. In 1994, Dr. Brunkow joined Darwin Molecular Corporation, a gene discovery biotechnology startup company in Bothell, Washington State. It was co-founded by Leroy Hood. Brunkow remained at Darwin after it was acquired first by Chiroscience and then by Celltech. And she remained there until the site closed in 2003. A few years later, Dr. Brunkow joined the Institute for Systems Biology in Seattle, and where she is still working as a program manager in genetics, and she coordinates multidisciplinary projects that integrate human genetics with whole genome sequencing and computational approaches with the overall aim to understand complex diseases and make discoveries of medical importance. She's currently actively working to promote education and access to education and training for young women through mentorships. And hopefully this platform that she now has will help her in these pursuits as well. So please join me to welcome Dr. Mary Brunkow to the stage to give her Nobel Lecture 2025.
Thanks very much for that introduction, and thank you all for being here. I especially want to thank the Nobel Assembly and Nobel Committee of the Karolinska Institute for this great honor. In the past couple months since the announcement of the Nobel Prize, I've had a lot of time to think back on the work that we did 25 to 30 years ago. And I realize that I wouldn't be standing here today if it weren't for a couple of really important mentors in my life who showed me what a life in research would look like. And the The first one of these is Dr. Larry Sandler, who was a fruit fly geneticist in the genetics department at University of Washington, where I was an undergraduate. And I was already pretty excited about genetics when I was in school there, but when I attended an upper-level genetics course with Dr. Sandler, I was especially enthralled by the way he wove Drosophila genetics lore into whatever topic he was covering in his lectures. And then I was really fortunate to be able to do an undergraduate research project in his lab. I spent about 1.5 years in his lab, where I saw what being in a research environment was like. And research was not really anything that I had on my radar as being a potential career track. I thought I wanted to go to medical school. But after spending a lot of time discussing with Dr. Sandler what my future might bring, I ended up going off to graduate school at Princeton University, where I was very fortunate to do my PhD research with Dr. Shirley Tillman, who is an extraordinary scientist and a fabulous role model, both for young women in science, but really anybody who's in her orbit. And working in Shirley's lab really taught me how to think critically, but also keep an open mind in science and in life. So I'm also really lucky today to have been preceded by the world's expert in immune tolerance, so I'm not going to spend very much time introducing the topic, just give a quick overview to remind you that immune tolerance is the process by which the immune system keeps a balance between T cells that recognize non-self antigens that would be harmful to the organism. These antigens could be presented by pathogens or environmental pollutants, and they are recognized and neutralized by the system. But the immune system also has to be able to tolerate self-antigens and not destroy the resident tissues in the organism. When this system becomes unbalanced, you see things like an autoimmune disease, and the resulting tissue destruction, et cetera, as the T cells attack self tissues. When we entered this area of research, it wasn't necessarily to contribute to understanding of immune tolerance, but we were more generally interested in identifying targets that would help us develop drugs to treat things like autoimmune disease or cancer or transplant rejection. And the vision that we were starting with was using genetics as a way of getting new insight into this particular biological area of research. And the story starts with this mutant mouse that we were interested in. And it begins in the Oak Ridge National Laboratory, which was established in Tennessee in 1943 as part of the Manhattan Project during World War II. So while most of the efforts in the laboratory were focused on developing the atomic bomb, there was also quite a bit of interest in understanding how radiation affects people and other mammals. And so a mammalian genetics section was established. And William and Leanne Russell, who were really top-notch mouse geneticists from the Jackson Laboratory, were recruited to Oak Ridge to head up this unit, where they were tasked with studying the hereditary effects of radiation and radiation-induced mutation rates. So the Russells did some really important experiments that did, in fact, contribute to our knowledge about the effects of radiation. But kind of a bonus for them as mouse geneticists is that they ended up with this huge collection of really interesting mutant mice, which over the years has been a tremendous resource for the scientific community in identifying new genes that are important in a number of different biological areas. So in 1949, they observed this mouse mutant that they called scurfy, mostly just because of the way it appeared. And interestingly, this is a spontaneous mutation that actually didn't arise in any cohort of mice that were exposed to radiation. And they maintained this strain for about 40 years before anyone really was able to study it in any depth. They just knew that it was something important because it has this really striking lymphoproliferative disorder. It's also the first X-linked mutation reported in mice. So just when you look at these mice, so on the left is a scurfy mouse, so a male mouse with a single X chromosome that's carrying the scurfy mutation. They show this kind of general wasting phenotype and a scaly skin that looks a lot like psoriasis or eczema. When you look internally, there's huge enlargement of the spleen, liver, lymph nodes, and this is due to infiltration by lymphocytes. The little scurfy mouse is sitting next to his sister, who is a carrier for the scurfy mutation, but there's really no phenotype in females. So in the 1980s, Virginia Godfrey and Irby Wilkinson came to Oak Ridge, and they performed kind of the first studies of the phenotype in scurfy mice. And they were able to show that this phenotype is mediated by CD4 positive T cells. They showed a marked cytokine elevation and concluded that this autoimmune syndrome is a result of a defect in the downregulation of T cell activation. So it seems to be really an incredible model for autoimmune disease. Now, fast forward to the 1990s, and what was happening at that time was kind of an explosion in new approaches and technologies and computational methods that were going to allow us to characterize the whole genome. So looking at all genes, gene structure, chromosome structure. The Human Genome Project was launched in 1990. And in 1993, this small biotech startup company, Darwin Molecular, was launched just outside Seattle, Washington. And the vision behind Darwin was to use genes as a way of discovering novel drug targets. And the initial areas of interest were in autoimmune disease, cancer, and AIDS. And interestingly, so I'm showing, let's see, these are the founders of the company. And David Gallus, immediately prior to coming to Darwin, moving to Seattle, he had been the director of biological and environmental science at the Department of Energy, which was the major funder for Oak Ridge National Lab. So he was very familiar with the mammalian genetics unit and well acquainted with the Russells, the geneticists, and he had a real appreciation for the power of mouse genetics in helping us really dive into particular biological areas. So David facilitated collaboration with the Oak Ridge group, and we decided to work first on figuring out what the molecular basis of the scurfy mutation is. And being a startup company, there was definitely that feeling of, you know, everybody wears multiple hats. Here's a picture of David pushing a broom through a lab. He was also the president and chief science officer of the company. So I joined the company in 1994, and my background was in genetics, genomics, molecular biology, and I was tasked with finding the mutation that causes the scurfy phenotype. And we decided to take a two-pronged approach to cloning scurfy that would involve first genetic mapping. So we knew the scurfy locus was on the X chromosome, and some sort of general localization had been performed already, so we had a good starting point. But I decided that we would just continue to let the mouse do the work for us and start trying to narrow that interval further by scoring for recombination events that would help to narrow the scurfy locus. So we brought the scurfy mice to Darwin in Seattle, but first we had to find a home for them, and so that was an underutilized janitor's closet with a sink and a self-contained mouse caging system. And I was able to set up a big backcross experiment in that janitor's closet in order to score recombination events. And this, oops, sorry. Oh. I don't know if I'm going backwards. Sorry, this is just a copy of a piece of paper that I had up on my bulletin board for years, it seemed like, where I was tracking recombination events and also trying to compare our X chromosome mapping to other maps that were being published at the time. It was a very confusing time where there wasn't, you have to understand that there was not really genomic sequence available at the time, and so but everybody was really trying to put together maps showing orders of markers, etc. So we combined the genetic mapping with physical mapping. So we wanted to ultimately really understand at the DNA level, at the nucleotide level, what was happening in the region that contained the scurfy locus. And as I said, as sort of an outcome of the Human Genome Project, there were a number of really, cutting-edge technologies that were available, and some of these are listed here. And I think it's worth pointing out that our situation was perhaps unique. Being in a biotech company, we had the funding that allowed us to incorporate a lot of these different resources, as well as the mouse genetic breeding experiment as well, which is all very expensive. It would be difficult for an academic lab to do all of this simultaneously. This was my core team that worked closely with me. Eric and Catherine were really very talented molecular biologists, worked great in the lab. Brian Paper was a very talented DNA analyst and bioinformaticist who had a lot of experience with both staying on top of what DNA sequences were available in the databases, which was pretty limited at that time, but also being really aware of new analytical tools that were coming online. So the process that we went through for finding the scurfy mutation involved, as I said, the genetic mapping. We ultimately found, so the red X's on this chromosomal region represent the most informative recombination breakpoints that we identified. And at the same time that we were doing the genetic mapping, we were using whatever molecular markers we had in that region to start pulling out large cloned fragments of genomic DNA and kind of generating a tiling path or a contig of artificial chromosomes across the region. And in the end, we were able to estimate that the physical region was about 500,000 base pairs long. And once we had a number of cloned sequences across the region, then we started doing really high quality DNA sequencing of them. So we had a really good core facility to do the DNA sequencing ourselves in-house. Then once we had that really high quality genome sequence across the region, we were able to apply whatever kind of analytical tools and algorithms that were available at the time. And we ended up with a list of about 20 genes in the region. So just by blast analysis, you could find homology to known genes for certain. So about half of our genes in the region were previously reported genes. But then tools like GenScan were actually quite good at predicting novel coding sequences. So they would spit out, exon intron structure and find 5' transcription start sites and the like. And this is just what the output from GenScan looked like when you fed it, a couple hundred KB of genomic sequence. So we had a list of 20 genes, like I said, and then the trick was just to sequence, and this was all sequence, of course, that was derived from the wild type version of genomic DNA. And so for each of those genes, we cloned and sequenced the scurfy version of the gene, looking for a mutation. And where we found the mutation turns out to be this gene here that we were calling FKH. It was a novel gene that had never been reported, but it did have homology to a family of fork head domain containing DNA binding proteins. And this is a page out of Catherine's lab notebook that shows that reports the mutation. I'm not going backwards very well. But anyway, so this slide here is kind of our overall summary then of the region, which shows the gene content. Again, these X's represent the most informative recombination breakpoints. And then I wanted to show this nice sequencing chromatogram, because we never look at those anymore. But this was a really nice example of that clearly shows an insertion of 2 extra adenines in the scurfy versus wild type. And this mutation, of course, would cause a frame shift in the translation of the mRNA. And it's located in the DNA binding domain of this novel fork head protein. So as soon as we sequenced as soon as we figured that we had the scurfy mutation, that big celebration all around, but then we realized that there was going to be quite a bit of validation and confirmation of this. But we also, at the same time, right away cloned and sequenced the human ortholog of this sequence, this novel sequence. And then we submitted those sequences to a nomenclature committee because the protein was part of this known fork head domain family. This is what was expected. At the time, you would submit sequences and the committee would do a phylogenetic analysis to determine where within the family this novel sequence lies. So then the FOXP3 name was assigned to our sequence based on where it fits into the whole tree of fork head domain containing proteins. One of the ways that we confirmed that we had indeed cloned the scurfy gene and that FOXP3 was scurfy was to use a transgenic mouse approach to see if we could rescue the scurfy phenotype if we provided the wild type version of the gene. So we subcloned a large genomic fragment that had only our novel coding sequence on it, along with some 5' and 3' sequence, with the hope that this transgene then would mimic or recreate the same expression pattern that the endogenous FOXP3 gene has. And so this purified DNA was microinjected into mouse oocytes, and we generated 5 independent lines of transgenic mice carrying the wild type version of the FOXP3 gene. And in those 5 lines, there was quite a wide variety of copy number, which was interesting because those overexpressing transgenic mice also have a phenotype, which Dr. Ramsdell will get into in more detail in a moment. But what what we found, well, first of all, what we found was that male mice that carried the scurfy mutation but also had the transgene, so we bred the transgene onto the scurfy background, and those male mice were completely healthy. They had, normal lifespan. You couldn't tell they had any phenotype at all, which was also strengthened by what we saw internally looking at organs. And so this just shows what a scurfy lymph node normally looks like with the great infiltration of lymphocytes compared to wild type. And you can see that when you combine the transgene and the scurfy mutation, they look basically like wild type. So we were really convinced by this that we had, in fact, cloned the scurfy gene. So we wrote that up and reported those results. But then we also were aware that there was a human condition that also mapped to a similar region on the X chromosome and shared a lot of features in its presentation with what a scurfy mouse looked like. And we collaborated with a couple different clinical genetics groups. So Hans Och's group at the University of Washington, and also Bob Wilden in the Oregon Health Sciences University. They had been studying families that have this IPEX syndrome. And we were able to get DNA from them, from the patients and families that they had been studying, and we indeed found that the human ortholog of FOXP3 was mutated in this condition. So we could demonstrate that FOXP3 is the underlying basis of IPEX, and IPEX is an acronym for all these different syndromes. And so basically, again, a very severe autoimmune syndrome in boys, in young boys. It's usually seen very soon after birth. And interestingly, in contrast to what we have from the mouse, where we had just a single mutation in the scurfy, with IPEX, now there have been dozens of mutations reported, which kind of gives you a nice allelic series in the gene, and mutations of all different types that affect different portions of the protein. And it has been reported that there's a pretty good correlation between the type of mutation in the gene and the severity of the disease. So there's a lot of variability in the way that IPEX presents itself. And this is significant because, well, first of all, these days, as soon as a child is suspected to have IPEX, the FOXP3 gene is sequenced. Which gives the clinician a lot of information about whether, it's an important diagnostic feature. And then, like I just said, since there is some correlation between the type of mutation and the severity of the disease, that also gives the clinician some prognostic information. And from a molecular biology point of view, all these different mutations really have helped people analyze in great detail what the importance of different functional domains of the protein are based on what these mutations are doing. So as I mentioned, FOXP3 is a DNA binding protein, and it is a transcription factor that controls a lot of downstream genes. It's a novel member of this fork-head winged helix family of proteins, highly conserved across all vertebrates, and it can act as either a repressor or activator depending on the other proteins that it interacts with. This is a very simplistic cartoon just to highlight some of the main functional domains in the protein. So I wanted to talk a minute about kind of what this means from a drug development point of view. So remember, we were working in a biotech company where the ultimate goal is to develop a drug that can be used in the clinic and help patients. And I kind of see two different scenarios coming out of an approach that, like what we were taking, where we started with an interesting phenotype. We want to discover a gene, but we have no preconceived notion about what the function of that, or what that gene or protein is going to be when we first start this kind of a study. So there's kind of two different scenarios that can come out of that. And the first is where the gene that is discovered doesn't really have direct human health implications. And what I mean by that is that the protein is maybe not particularly druggable itself. And FOXP3 is a good example of that. Being a DNA binding protein, it's in the nucleus. It's pretty hard to access with, a small molecule or a biologic type drug. But I'll get back to you. I mean, it doesn't make FOXP3 any less interesting, but it is hard from a drug development point of view to figure out how you could directly manipulate FOXP3. The second scenario is one in which the gene discovery that you make actually does lead to a protein that is accessible and a good drug target. And I just want to take a short detour and describe a project that I was also involved in that was happening in parallel with the Scurfy project. And this is one where we discovered a gene called SOST. And in this case, what we started with was a human condition called sclerostiosis, which has a very strong effect on bone density and bone growth. It's an autosomal recessive, so presumably a loss of function of some important gene. And what's shown here are x-rays of skull and hands from a normal adult compared to what you see in an individual affected by sclerostiosis. And you see this, really tremendously increased bone density. It's characterized by excess deposition of bone throughout life. It's very strong, as in contrast to some other conditions that affect bone density. The quality of the bone is actually very high, and all different bone types are affected. And there's very little other pathology that's not bone-related in this condition. And carriers, interestingly, so if you're heterozygous for sclerostiosis, there's really no apparent visible phenotype, but they do have an intermediate bone density, which implies kind of a dose-dependent, which is very nice from a drug development point of view also. So we went through a similar positional cloning exercise as what I just described for Scurfy, but in human families affected by sclerostiosis. And we, at Darwin, at the time, we were kind of working under this, this idea of a surrogate drug approach. And what I mean by that is that we were specifically looking for phenotypes in mouse or human which under certain conditions would be a clinically relevant phenotype that you might want to actually use to treat a disease. So what I mean by that in this case is that in sclerostiosis, this loss of function mutation is causing a big increase in bone density, which would be exactly what you would want to do in an adult that's affected by low bone density. So like in the case of osteoporosis or chemotherapy-related bone loss, if you could kickstart bone development in an adult, perhaps you could, really increase their quality of life. And so at the end of our positional cloning effort, we identified a gene called SOST. And this was very exciting on a number of levels. First of all, it was a novel gene, which was great for the company from an intellectual property point of view. It's secreted, which does in fact make it really accessible to small molecule or biologic type drug that could actually interact with this gene product and modulate its effect. And it's working in a biological system that was already fairly well understood. So it's an antagonist of BMPs, or bone morphogenic proteins. So it was an area that where there was already a lot of knowledge. So this was an example of a really successful outcome from this kind of gene-based drug target discovery approach that the company was doing. But back to FOXP3. Oh, and the most important point, of course, is that there is, in fact, a blocking antibody now that's on the market. There is actually a drug that's being used to treat osteoporosis, and this was co-developed by Celtec and Amgen. So back to FOXP3, and I'm pretty much wrapping up here. I want to just summarize some of the things that we know about FOXP3. And to get back to the point of that FOXP3 itself as a protein is a hard one to approach from a drug development point of view, but it being a transcription factor, in fact, opens the whole world of downstream targets and other genes that that regulate FOXP3 itself. And in fact, once this work was published, there's many genes or many labs around the world who have in fact made great progress in understanding the whole kind of gene regulatory network that FOXP3 works in. FOXP3, and this is kind of an extension to what Dr. Sakaguchi was talking about, we were able to show that, so we were aware of these suppressor or regulatory T cells that Dr. Sakaguchi had reported on, the CD4 positive, CD25 positive cells, and we were able to show that FOXP3 is really very specifically expressed in that very small subset of CD4 T cells that are also expressing CD25. And these really define the regulatory T cells that we've heard about today. So FOXP3 was a really important marker and kind of gave researchers a hook into this whole new field of regulatory T cells. We now know also that FOXP3 actually programs or directs the differentiation of CD4 positive T cells into the Treg pathway. And there are now a lot of interesting advances that are related to harnessing the power of the Treg cells, sort of separate from the FOXP3 protein itself, but the Treg cells that are defined by FOXP3. We've made a lot of progress in autoimmune disease, organ transplantation, and cancer. And again, you'll hear more about this from Dr. Ramsdell, which is exciting. Definitely a big team effort. So the folks at Darwin Cell Tech are listed over on the left with kind of our key team members in bold, but really there were so many people within the company who contributed at various levels to this study. Our collaborators at the Oak Ridge National Labs, and then the clinical geneticists at University of Washington and Oregon Health Sciences, we owe a lot to. And then, I obviously am not working with the people at Celltech Darwin anymore because the company doesn't exist anymore. The doors closed in 2004, but I have been working at Institute for Systems Biology for a number of years, and I must say that, since the announcement of the Nobel Prize, I've gotten just, loads of support and encouragement from people at ISB, and I just want to call out especially Jim Heath and Lee Hood, who have really helped a lot, sort of navigate this new life that's happened since two months ago. And just as a quick, indicator of how close-knit the team was at Darwin Cell Tech, a couple weeks after the Nobel Prize announcement, this big email went out to every former employee that we could contact, and we had a reunion, pretty, short notice at a bar in Seattle, and you can see there was great enthusiasm and quite a good turnout of people showing up to celebrate this great news. So with that, thank you.
It is now my great pleasure to introduce our third Nobel laureate, Dr. Fred Ramsdell. Dr. Ramsdell was born in Elmhurst, Illinois. He earned his Bachelor's of Science degree in Biochemistry and Cell Biology from the University of California, San Diego. He then moved up the coast and did his PhD at the University of California, Los Angeles. and graduated from there in 1987. Following this, he transitioned to the NIH, where he performed his postdoctoral training, and he focused there on immunology and specifically immune tolerance. He then had a stint of working at a number of companies, first at Immunex, and in 1993 he joined Darwin Molecular Corporation around the same time as Dr. Mary Brunkow, as you just heard. At Darwin, he was the person who established the immunology program, and which then led to the groundbreaking work that were about to come. Between 2004 and 2008, Dr. Ramsdell joined and worked in many companies, including Zyme Genetics. He established the Inflammation Research Center in Seattle, which was part of the Novo Nordisk Seattle site. He also worked for a while at Atar Pharma, and then he was a chief scientific officer at the Parker Institute for Cancer Immunotherapy in San Francisco. Following this, he co-founded Sonoma Biotherapeutics, which is a company that now works and focused on the development of regulatory T cells as therapies. And this company was co-founded with Jeffrey Bluestone, Xixing Tang, and Alexander Rudensky. And he was the chief scientific officer there until just recently. So for our last speaker this afternoon, it's my great pleasure to introduce Dr. Fred Ramsdell to the stage to give his Nobel lecture.
Thank you very much for the kind introduction, and I want to thank the Nobel Foundation and particularly the Karolinska Institute, the staff and faculty here who have been absolutely fantastic. And I think all three of us as laureates consider this to be an amazing award, and it's a very humbling experience. But I think we're learning to enjoy it finally. I'll throw some disclosures up. That's pretty quick. I'm going to divert and not talk about this for a minute because this really has been a very amazing week, and it's only 2 days old. But nonetheless, we were at the Nobel Museum for the last couple of days, and you may or may not know that they asked designers to design a dress for each one of the disciplines every year. And this is the dress for physiology and medicine this year. And the dress is called U, Y-O-U. And it's designed by two young women from Beckman's design group, Odessa Galloway and Lovisa Schmidt, who I did not get to meet, but I would love to one day, because when I looked at this from the back, I immediately saw this interesting pattern, which only a T-cell dork like myself would really notice, which is that if you look at the part of the dress here that kind of comes up and it's got a long train, and this is the Y part of the U. Okay, there's a little bit of this is a mannequin, so there's a little bit of midriff there, and then there's this circle and this other shape above that circle. And so I looked at that and said, this looks exactly like a T-cell receptor binding to an MHC molecule that has a peptide in its groove. And the very lovely woman at the Nobel Museum said, that's exactly right. And I thought, how in the world do you know that? That's incredible. And more importantly, how do these two people know that? That's incredible. I still don't know the answer to that, but I'd love to find out. But if you looked at the diagrams on the back, that was clearly exactly what they were trying to do. So I was incredibly impressed. So even the dress designers in Stockholm apparently know an awful lot about immunology. All right, so we'll come back to why I'm actually here. It's not to talk about dresses, which is the scurfy mouse and how that's led. And I'm going to tell a little bit of a story and bring us kind of full circle into where we are literally, at the moment in terms of how we're trying to advance these programs. So this is a timeline that was put together by my friend and former boss, Jeff Bluestone, Megan Levings, and Victoria Fung at UBC. And whoa, I gotta go back again. Sorry, Mary. Okay, Mary, got it. All right, so we're gonna, dang it. I knew I was going to do this. All right, so we're going to go back to the early 40s and 50s and 60s. And in 1960, there was an award to McFarland Burnett and oh my God, how am I blanking? Whoever else is listed up there. For the concept of peripheral tolerance. That happens to be the year I was born. So 65 years ago, the Nobel Committee awarded this Nobel Prize for the concept of peripheral tolerance and the ability of people to control responses to their own tissues. So we're going to eventually fast forward 65 years to today, where another Nobel Award recognizes the same thing, which is pretty amazing. It's either amazingly quick or amazingly slow, depending on how impatient you are. I consider it slow because I'm pretty impatient. But I'll just tell you why we're working on this again and remind you of this. And this is data from Lisa Clark, who was in the lab at the time. And when we got the scurfy mice from Oak Ridge National Labs, and Mary has talked about this, as has Shimon previously, this is just some of the data that we generated. These mice, in many, many different ways, were hyperactive. If you took the lymphocytes out of these animals, the T cells out of these animals, they responded incredibly quickly. Normal cells need 2 signals. So this is shown over here. Normal cells need 2 signals to give you a good response. This is looking at those cells' ability to divide. This is to make cytokines. It's basically the same answer. Scurfy cells don't need that second signal. They divide like crazy, and they don't seem to care, which was fascinating to us. So when we generated the transgenic mice, and Mary has told you all about the cloning of this gene and the fact that it was the last gene in the list of possibilities, I'll come back to that, was a little distressing to us at the time, or at least certain to me. But when we cloned the gene, we also made these transgenic mice that Mary talked about. So this is another example of again, stimulating these lymphocytes out of these animals, and this is more stimulation, less stimulation, more response, no response. Normal animals look like this. The mutant animals, again, hyperactive, but these transgenic animals were hypoactive in every assay we looked at. And as she said, we had 5 different lines of mice. The more of the scurfin protein that was present, the less responsive these cells were. So it was the yin and the yang of responsiveness, and it was I called it a rheostat for functionality at this particular point in time. And we didn't know what that meant, but we thought that was a really good hook into how we might use these cells. And we also were aware of the really pioneering work from Shimon and his colleagues published here, but we always cite this one paper, but there's more than one paper. There's a ton of work looking at this small population of cells in the mouse that had CD25 and CD4, and Shimon's described these quite well. Roly Kotri in the lab did this experiment very early on and showed that if you isolated just these cells, those are the only cells in the hematopoietic system within the T cell lineage or any other lineage that express the FOXP3 gene. So that, to me, was a revelation. So that obviously provided a molecular marker for what these cells were, but it also meant that this gene, at least it meant to me, this gene drove these cells and was really responsible for that particular lineage. It was also responsible for their functionality. So this is another way of showing some data that Shimon's already shown in principle, which is when you put normal T cells into a system, you can stimulate them and they'll give you a response. If you add regulatory T cells, so these Shimon Treg cells, it will inhibit that. If you add non-regulatory T cells, you get an increased response. We knew that. That was perfectly predictable. When we looked at the transgenic cells, however, again, the regulatory cells from these transgenic mice were very good at inhibiting, but now these cells which don't normally have suppressive function now do. So again, FOXP3 expression in any T cell can drive at least some aspects of a regulatory program and a suppressive program, which was amazing to us. So we also knew that these cells are derived from the thymus, and we asked, well, what happens if we only express this gene within the thymus? And so when the cells then leave the thymus and go out to the periphery, what will happen in that case? So we did that experiment, and it turns out expression in the thymus gives you thymic Tregs, but it does not prevent disease when you cross these animals, that gene onto scurfy mice. So this, and forgive the nomenclature, but this is the transgenic mouse you've seen before. So if it has the transgene, the normal FOXP3 gene, you don't get any disease. But if it has that same gene, same FOXP3 gene that's only expressed in the thymus, these animals still come down with disease. It's the same way scurfy does. So expression in the thymus and programming and driving the initial differentiation of these cells is not sufficient. You need to have this gene on in the periphery of these animals. This is shown in a very elegant, though complicated experiment by a good friend and colleague, Sasha Radinsky, much later, but he was able to turn on and turn off the FOXP3 gene genetically within these mice. And what he could do is allow the animals to develop without FOXP3, which means they developed the scurfy-like phenotype. But if he turned FOXP3 on in Tregs at 4 weeks of age, those animals then reverted essentially to a normal phenotype. You can see here they're not totally normal at this particular point in time, but inflammation goes down dramatically, and those mice will continue to live. So this really is a dominant form of tolerance in the periphery that can control an ongoing immune response, which is what we need to do in the context of disease therapy. So we were pretty confident at this point that FOXP3 was the master regulator for T-cell development. We certainly had that data. Shimon had that data. And quite quickly, many other people in the world reproduced basically the same essential findings, which was great. It's a necessary gene, and the absence results in a hyperactive immune response, which ultimately results in lethality, whether it's in a mouse or a person, without dramatic intervention. So that was great, and we thought this is a really, really good concept for how we can control the immune system. But, as Mary pointed out, this is a terrible target for therapeutic development. In the year 2000, there was no way to target a transcription factor that made any sense. Even though we tried, we failed miserably at it. And there's no way we're going to actually turn this into a drug. We certainly thought about lots of ways we could either enhance regulatory T-cell number or function, and some of these are shown here. Some of them work to a first approximation, but really they're not therapeutically viable for the most part at the moment. There's some caveats to that, and there's ongoing work still, but in general, this wasn't really a very fruitful approach back in the year 2000, 2005. We also knew on the tumor side, and Shimon talked about this, that if we could eliminate or at least inactivate these cells in the context of a tumor, we could potentially drive good tumor responses. And certainly that's been shown in the mouse. We think that will happen in the human, and there's ongoing clinical studies, which I won't talk about right now, to try to do that in human tumor settings as well. And we'll see where that ends up in a few years. So I'll come back to this. But because we couldn't target this in a conventional way from a drug perspective, we essentially said, okay, we need to finally get this published and get this out there, and everyone else can work on this and hopefully help us figure this out, which is what we did. And lots of people have helped us out over the years. And in the intervening decade, we learned a lot about how these cells work. There's multiple ways in which they can control an immune response. They can suck up the growth factors from the environment. They can modify the metabolic environment around them. They can deactivate antigen-presenting cells by literally sucking proteins off the membranes of those cells. There's a number of other ways that these cells can control immune responses, and that is almost certainly context dependent on the disease and the tissue, et cetera. We've also learned that these cells are resident through every tissue in our body. In fact, one of the reasons it was the last gene we prioritized to sequence was because the data we had suggested that this gene, this FKH gene, as Mary called it, was expressed in every tissue in our body at a very low level. And I thought that was stupid. That can't be what's causing this disease, so that can't be it. Of course, completely wrong. These cells are resident in every tissue in our body, and they're really important for maintaining normal homeostasis of our tissues. So these cells not only suppress immune responses, which they're very effective at, and they home very well to inflamed sites, but they also have tissue repair capacity. They will help regenerate tissue. They will help repair damage from an inflammatory response, et cetera. So they're somewhat like having both the police at the site of an accident or at the site of an event, stopping whatever bad is going on, as well as having the medics there who are stopping the bleeding and keeping people alive. So they do a little bit of both functions. And so we've learned a lot about these cells over the years. We've actually learned how to identify them and actually get a good handle on them in the human. You know, all of the data you've seen so far is using the CD25 marker in the mouse, which is very, very clean, works great. Doesn't work at all in the human because we're more complicated than mice, surprisingly. So we did find ways to isolate those. Jeff Bluestone and Kizhi Tong and others at UCSF identified ways to actually isolate human regulatory T cells. And that happened in the late 2000s, and that enabled some very early studies in which those isolated cells were put into the lab, grown up, and given back to patients. And when that happened, patients were fine, they were safe, they migrated to sites of inflammation. You could see biomarkers of activity as though these cells were doing something, but there wasn't really a meaningful clinical benefit for most of those patients. And so, okay, the simple version of more will fix the problem may not be the correct answer. Again, there's some caveats around that, but in general, it looked like more isn't going to be enough. We're going to have to do something else to these cells in these patients. And so in 2019, right before the pandemic came along, Jeff was convincing in that he could start a biotech company, and Tong and Sasha and myself basically supported Jeff, and I joined Jeff in the company, and we started Sonoma Biotherapeutics. And we really did this because we liked taking advantage of the fact that this is nature's way of controlling immune responses. These are evolutionarily defined cells that have been created over, you know, thousands of years of evolution, and their job is to control autoimmunity. So essentially, I don't have to be smart. I just have to use the right cell. And that works perfectly for me in terms of my ability to predict things. So we're using the right cell, we believe. They'll block inflammation. They can inhibit ongoing inflammation, and they can repair tissue, all in one drug. So for us, having this multifunctionality within one drug made a lot of sense, as opposed to some of the other therapeutic approaches that we're doing. So, and by the way, there's numerous companies and other organizations that are actually doing very similar things. I'm going to talk about Sonoma because it's the one I know the best, but there's many other groups that are doing the same sorts of things with their own little twist in different areas. And we can talk about those later if you want at the reception afterwards. So we started with rheumatoid arthritis. There's a number of reasons for that. One, there's great preclinical data to suggest that regulatory T cells can reverse as well as block rheumatoid arthritis in mice. Now, the mouse models are far from perfect, but at least that's a good start. We could show that in arthritis patients, the regulatory T cells don't look as good as they do in healthy volunteers. And there's a number of ways we can show this. It's not that they're absent, they're clearly there, but they're not as active as they are in several assays in human patients. And in fact, the worse you are in an arthritis score, so which is to the right here in this graph, the less good your regulatory T cells look to some extent. Okay, so we thought, all right, they're slightly defective. Can we use those cells, take them into the lab, and make them better and put them back in? Because we think, you know, those cells can be resurrected, and there's a whole bunch of data suggesting that would be true, and then we could treat patients with them. We can expand these cells in the lab. This is normal. This is RA patients, again, not quite as well, but certainly good. And the cells look like very good regulatory T cells when we get done expanding these cells. So that's terrific. But what are we going to do to make these cells better? Why are they going to work in arthritis patients? And so we decided we would actually give these cells specificity. So normally, regulatory T cells have a very diverse repertoire of T cells, and some are specific for whatever it is you're interested in. But we wanted to make a lot of ours specific for something that was found in the rheumatoid joint. And so what we chose to make our cells specific to are citrulline proteins. And so without getting into the details, these are modified proteins, and they're modified during the process of inflammation. And so in an inflamed joint, there are a lot of these proteins that are generated, and they sit in the synovial fluid and in the synovial lining of an inflamed joint. And it's well known, and it's well known largely from work actually from the Karolinska. And this is just an example of showing that there are a lot of these citrulline proteins within an inflamed joint in an arthritis patient. So we decided we would make a CAR. So this is a chimeric antigen receptor. We would give specificity to our Treg cells. And this is essentially stealing an idea from our oncology colleagues. Clearly, this is a thing that works to treat a variety of, right now, liquid tumors. It's very efficient, but the technology to generate these is actually fairly robust. And so the way you make these is you take a part of an antibody, and you stick it on a part of a signaling domain, and then you stick it, in this case, in a regulatory T cell. Normally, you put these in other cells to kill tumors. In our case, we're just going to focus this regulatory T cell, in this case, on citrulline proteins. And we did this as a collaboration with Lars Klarsgood and Vivian Mellstrom here at the Karolinska, because really, they are the experts in this. And they have hundreds of these antibodies that were derived from patients. And we selected one that saw a variety of these different proteins with good affinity, good activity, good specificity, et cetera. So the binding part, the business end of our CAR, essentially comes from the Karolinska Institute. Okay, and what you can show is that that CAR Treg now sees things like synovial fluid. It certainly sees purified citrulline proteins, these modified proteins, sees them quite well, a variety of them, not a problem. It responds to them and does everything a regulatory T cell is supposed to do. But it also sees synovial fluid coming out of RA patients, which was really important to us. We wanted to know that these modified proteins that are found in disease, in the tissue, was going to be able to stimulate our drug product, and in fact, it does. So this drug product, again, it's a regulatory T cell, so it's the whole cell. They all express FOXP3 after we've grown them up, and I'll share the protocol for that in a second. So they all look like good regulatory T cells by the fact that they express FOXP3. There's other markers as well. This is a little gene expression analysis. I'm not going to walk through this, but the first two columns, in fact, are our drug product. The two middle columns are, in fact, a non-CAR-bearing version of the same thing. And all these red ones on the right are effector T cells. So this is the opposite side of the equation. And so you can see that our regulatory T cells are completely different from these activated, potentially pathogenic cells. So that's not what we're making when we make our product. We did design a manufacturing process for this. And so what we do is we take blood out of a patient. We isolate the regulatory T cells out of that blood, which is a very long process, takes us essentially all day. We put those cells into culture, and we transduce them with a virus that puts the CAR into those cells, and we grow them up for 14 days. And we get billions of cells at the end of those 14 days. They're frozen, quality control checks are done on them, and then they're reinfused back into the patients. And I'll show you some clinical data on this in just a minute. It's a long process, and it's an interesting, challenging process, but it is one that you actually can do. And again, our oncology colleagues have shown it works in that setting. It's not really fundamentally that different here, and so we're working on the backs of the oncologies. And this really just shows that the cells that we get are in fact, if anything, more Treg-like. So this is at day 14 after they've been grown up, and this is a bunch of markers for Tregness. This first one is FOXP3. They're, if anything, more Treg-like after the 14 days than they were going into the culture. So we feel pretty good that these cells are stable and that they're effective, and they will be effective when they get into patients. So this is the clinical trial that's ongoing right now. It's a phase 1 trial, so it's a safety trial. And I can say that so far, every patient who's been dosed is fine. There are no significant adverse events yet. Now, we've done 2 dose levels, so we've done low and middle, and it's only 3 patients each, so it's 6 patients total. So I certainly don't want to overstate the significance of this, but I'll show you the data in a second, and I think this is how we're trying to prove that these regulatory T cells will, in fact, modify disease in a patient population. These are the patients. And one of the things that's interesting I'll highlight down here on these patients is that every patient, so these are the 6 patients I'll show you the data on, every one of these patients has had at least 4 different types of therapeutic intervention. So they've had JAK inhibitors, they've had anti-TNFs, they've had other things, at least 4 different ones, none of which have been effective for these patients. And in some cases, this patient's had 12 different drugs. They've had disease for 28 years. They've gone through 12 different drugs. Their disease continues to progress. So these are patients who, in many cases, are very refractory to the existing therapies that we have out there. So it's a pretty high bar at this particular point to study these patients. So this is the meat slide, if you will, which is on the left here is the axis of it's either tender joints or swollen joints, depending on which one you're looking at. It's either 68 joints or 28 joints, doesn't really matter in this particular case. These yellowish things are the low-dose group of patients. The bluish things are the mid-dose group of patients. And what you can see in most of them is after the infusion of the product, there's a diminution in the score. So the patients get better. Not all of them, and clearly to varying levels. In some cases, the scores come back up over time. This is 24 weeks. In some cases, the scores come back up. In some cases, they come up a lot. In some cases, they stay down for 24 weeks. In some cases, it's in between. So very early days, but we're very encouraged by the fact that at least we're seeing some response in these patients. There's a lot of other measures that we're taking that I think are even sort of anecdotally useful. Patients can now open a jar, which they couldn't do previous 5 years. We've had patients who have gone for a run, which they haven't done in the previous 5 years. Again, that's all anecdotal. It's small, but I find it encouraging as well. When we look at as much as we can about mechanistic data and what's going on in these patients, this is one patient where we've done some imaging, some ultrasounds. And the top is 2 different joints, and you can see the red, a lot of inflammation. This is 4 weeks after infusion, a little bit of inflammation left, very little in this particular joint. So we can see inflammation dropping by a quantitative measure in this particular case. In some, but not all patients, we're getting biopsies. So this is the medium dose set of patients, and we've gotten a number of synovial biopsies from these patients. One before we gave the infusion, so pre, and 4 weeks after we gave the infusion, post. And this is a score of inflammation. This is the percent of inflammatory cells compared to all cells. And you can see that there's a reduction. In this particular case, a very dramatic reduction in the inflammatory score. 9 is the highest possible value on this particular scale. So it went from 9 to 1 or 2 in this particular scale. So very dramatic reduction in that particular patient. More modest than some of the other patients. But at least we're seeing a reduction in the inflammatory scores, which again, we find encouraging, certainly not proof yet, but enough to keep us moving forward in this kind of trial. We can also, in again, a small number of patients where we have the biopsies, you can't see this, I'm sure, but what these red arrows point to are our cells. So we can specifically look for the cells that express that CAR in this joint. We can find those cells. When we look at the characteristics of those cells, they look like good, regular Treg cells. They have high levels of FOXP3, they're CD4 cells, et cetera. So again, our cells have migrated into the joint, they're there, and they still look like good regulatory T cells. And so we're relatively hopeful that as we go to higher doses, we'll see even more effects. And so that's, again, ongoing, and we'll have a lot more data in a year, but I just thought it would be useful to show how we actually really have now come full circle. Back in the 40s and 50s, when this peripheral tolerance was proposed and a Nobel Prize was awarded in 1960, you can follow this through, 65 years later, there's another Nobel Prize for being able to understand how this process now happens. And relatively quickly, at some level, we're now actually into clinical trials. And again, it's not just Sonoma. There's a number of groups trying to do this with their own little twists on this. And so I think in the next few years, we're going to have a very good idea of how well this works, where it works, when it works, and what we might need to do to make this even more efficient. And again, for me, this is very exciting because we're using this natural cell that has all the properties it needs, in theory, to control disease. And so what we're hoping for is not a treatment. We don't want to treat disease. We actually want to cure disease by resetting people's immune system back to the state it should be in and the state most of us have. So it's a goal, it's aspirational, but I don't think it's unachievable at this particular point in time. So I'm going to thank some people as well. Mary's already commented about David Gallus. David was a terrific scientist. He was a great mentor, a great friend. He's really, truly a Renaissance man at many levels. I actually have his art hanging in my house. I have a book of his poetry. David was truly one of the fine gentlemen around. Obviously, Mary's contributions, I don't think any of us would be standing up here if Mary hadn't been incredibly talented and done what she's done. Steve Ziegler, who's at the Benaroya Research Institute, was the first person really, I think, to recognize the value of the scurvy mouse. And he and I had long conversations about this. And he said, shouldn't we work on this? And I said, a little expletive, yeah. And Rolly Contri, who's somewhere in the audience, is really the first person to know that this gene was the thing that controlled regulatory T cells. And she'll never get enough credit for that, but it was tremendous work on Rolly's part and her team, which was fantastic. I'll also acknowledge the Sonoma team. Again, Jeff, who really got Sonoma going. Sonoma wouldn't exist. This whole process wouldn't exist without Jeff. Josh Bialke was the first employee at Sonoma, and he's driven so much of the work around this particular RA program and gotten it into the point where it could go into the clinic. Malin runs the clinical program, and Samir is the project manager. There's way too many clinical investigators to name, but watching clinical investigators go from cautious and not quite sure how they wanted to buy into this to now being full bore into this and wanting to treat patients and recruiting patients faster than we can make them is pretty impressive. And of course, Lars and Vivian here at the Karolinska, incredibly great collaborators. I will also make a little acknowledgement of two earlier mentors. Sid Golub was my PhD advisor at UCLA, really taught me the concept of perspective and being able to focus on something important. And BJ Folks at the NIH, who was just an amazing PhD advisor. I was her first postdoc, and I think we both learned how to work together as a team, and it was a great joy to work in BJ's lab. I will also thank the mice. No mice, none of this story. I and many others will correctly point out that mice are not a great model for human disease, but they really are a pretty good model for the fundamental tenets of how the immune system works. And we've learned enormous from them over the years, and I just want to thank them for that. I shouldn't say this with the mouse picture up, but I also want to thank my partner in crime, or partner in misdemeanors, I'm not quite sure what that is, which is my wife, Laura. She's put up with my inquisitiveness and my contrarian nature for 40 years, mostly with good cheer. And I wouldn't be here if she wasn't metaphorically standing next to me. And Thomas is my last slide. I mean, I'm over. But I was here in 2002. And I've mentioned a bunch of times when I was wrong, which is not an uncommon trait for a scientist. If scientists are always right, they're either kidding themselves or they're lying. So I'm wrong a lot. And I was here in 2002, and this is more my normal state. I've got sandals on. Usually I'd have short pants, but it must have been a cold day. But we were wandering around the old town, and we came across this travel trailer, which looks a lot like the travel trailer I was in when I didn't get the phone call from the Nobel Committee. But this one is from the Nobel Museum. and I stood in front of it and said, look, this is as close as I'm ever going to get. Apparently, I was wrong. Thank you all.
Dear guests, the 2025 Nobel Lectures in Physiology or Medicine has now come to an end. I would like to extend our gratitude to the Nobel Laureates for their extensive presentations this afternoon. Thank you all for attending, and thank you for sharing this significant moment with us. When leaving the hall, please exit the same way you came in. Those of you continuing to the reception, please be prepared to present your ticket. The QR code will be scanned. And now, there is an opportunity to photograph the Nobel laureates on stage. May I ask the laureates onto the stage for a round of applause.